Merci pour tous ceux qui nous accompagnent aujourd'hui.
Well, thank you. Thank you for all those who are here today. I'm going to open the session, this afternoon session, on conditioning and addiction from the animal model to the clinic. And this made me smile because this is a kind of conversation that I have often with my colleagues, and they just ask me, how can we take an animal and translate all that into a human model? So we're going to talk about all this, and I will first invite Dr. Serge AHMED to join us. He's going to talk about the role of interoceptive conditioning in cocaine craving. Thank you very much, Bernard, for your introduction. Thank you all, and thanks for being here. I have not thanked the organizers of the Congress enough, so I would like to really insist in acknowledging the work of the organization of this Congress. Thank you, Jean-Pierre Arcaitz, for inviting me to take place in this symposium on conditioning. I'm going to tell you about the work that we've been developing in our lab about interoceptive conditioning and its role in triggering craving, mainly using an approach with animal models. But before I get into the subject, into the topic, I would like to remind you of what conditioning is from a historical point of view. And I'm going to remind you of the Pavlov conditioning. It's about Ivan Pavlov. I'm sure you know about this experience. It's an experience through which a dog starts salivating when he hears the bell ringing, and the bell ringing is associated to viewing a bowl of food, a palatable bowl of food. So what's important here is that the sound of the bell at the beginning does not have any effect in the salivary reaction of the dog. It's only because the dog starts associating the bell with viewing the bowl and probably smelling the food. It's only that makes a sound trigger salivation. So this experience was made in the 20s, but of course, a whole century has elapsed, and we've learned lots of things about this Pavlovian conditioning. And we know that today, what is really happening regarding this conditioning is that there is a neuronal connection which is directly established between the perception of the sound and the neuronal circuit that controls the salivary reaction. But on top of that, it's the sound of the bell that evokes a mental representation of that bowl of food. So this is the desirable object. So, and if we were allowed to see what's going on inside the head of that dog, when the bell rings, the visual cortex is activated. It's just as if the animal was seeing the bowl of food. So this is an experience that we have made as well with rodents. And in this case, well, it was another kind of food they like. So what we learned is that the conditioned response does not only end with a salivary response. Actually, when we see the dog, we can observe that, well, by the sound of the bell, the dog is activated, and the dog goes towards the place where the bowl should be. The dog may even bark. I mean, there are lots of behavioral signs that make us induce that desirable food. But what we know as well is that the view itself of that food, this is also a conditioned stimulus. But this has not been taken in the context of the lab experiment. This is something that the animal has learned himself. And this is a conditioned stimulus to the palatable food which is inside the bowl. I mean, it's when the dog sees the bowl that he guesses that there is some palatable food inside that bowl. And what we have discovered recently is that this is a conditioned stimulus that is much more inconscient. And we know today that after digestion, there are part of the sensor neurons in the vagus nerve which are stimulated, and then these neurons go stimulate the dopaminergic neurons, and this creates a conditioning, and it's that interoceptive process that takes part because it's inside the body, and this is what creates that crucial reinforcement that will trigger all this chain of conditioning that I just described to you, with the bell and the bowl of food and the salivation and the stimulation of sensory neurons and the vagus nerve. So we know that all this association, well, it's a great problem, actually, because you want to decondition all that. And all these associations, they depend on the context. So, for instance, in Pavlov's experience, the association of the sound of the bell and the triggering of all that sequence of behaviors and physiological effects that make us see that the dog wishes this palatable food, this is all within a context. If you remove the context, the association will probably disappear. So do not forget that this capacity is very much developed in human beings, and it's the capacity to contextualize a series of associations. So it depends very much on the context. If you change the context, it may not work anymore. So this was just a historical reminder that I wanted to show you, just to give you the general picture. And I would also like to remind you that conditioning of drug effects has also been discovered in the lab of Ivan Pavlov, and it was Dr. Krylov in the 20s who made the first experience of conditioning involving a drug. In this case, it was a subcutaneous injection of morphine and the salivary reaction in the individual. So as you all know, those conditioned stimuli play a major role in addiction, induction, triggering, evoking craving, that urge to use the substance. Now, here I was just giving you an example, but let me tell you about this experience. It's an experience involving alcohol-dependent people. So we present them with a glass containing alcohol, and this triggers craving compared to the other stimulus. So we know that these are conditioned stimuli which generate craving. And here we see a systematic study by Marc AURIACOMBE in Marc AURIACOMBE lab, and we see that this craving, that this conditioned craving, could lead to a reuse, to a relapse into using again. So this conditioned induction of craving may lead to another episode of use. And, well, this is where I want to interfere about interoceptive conditioning. It's once the individual has this craving that is conditioned by something exteroceptive, that is, for instance, seeing a glass, where this triggers the episode of alcohol use, it's the interoceptive effects, all the physiological changes that the drug creates from a peripheral point of view. It's all that generates a stimuli. And this is conditioned, and this will in turn trigger craving. As you know, even when a person takes a drug, well, he or she is under craving, and this craving will make the individual repeat the use. So that's why it's so important to study this conditioning phenomenon, because this involves interoceptive stimuli. And here in this slide, I'm going to show you a classical experience by Jérôme Chaffé, in which cocaine-dependent subjects receive an intravenous dose of cocaine, and this increases craving. And at the peak of craving, these people would be capable of paying a lot of money for another dose of drug, much more than if the effect of the drug was dissipated. So what I mean by all this is that there may be two routes to craving. One of them would be basically the treatment of the stimuli from an exteroceptive point of view and an interoceptive route. And both routes, the extero and interoceptive, would lead to a neuronal substrate of craving, which is located in the prefrontal cortex. So how do we know all of this? Well, the craving can be conditioned by exteroceptive conditioning, but also by interoceptive stimuli and generated by drugs, and by other interoceptive stimuli generated by something similar to the drug, such as some moods and states and stress. For instance, certain stresses are confused with the effects of drugs. And here, well, I don't want to go into detail, but yeah, sometimes they are mixed with drugs. So, how can we test that hypothesis, the importance of that interoceptive conditioning? Well, first of all, you need to have an animal model of craving, and this is what I'm describing here in this slide. This is a classical model. It's a range statement model, which was very much developed by James Stewart and Harriet DeWitt at the beginning of the 80s. And according to this model, well, you have one first stage, which is about the acquisition of self-administration behavior, and there can be a scale-up phase and then prolonged access. And then there is another phase where animals take 2 intravenous doses of cocaine, and once they have developed that behavior and that scale-up of consumption, then you move to another phase, which is extinction. And in this case, the animals may push the button, but they will not get the injection. And at the beginning, they just insist in pressing, but they don't get any dose of drug, and they end up stopping. And at that time, the animal being naive, the doctor will still provide a dose, even though there is no association in this case with the stimulus. And then what you observe by doing so is that there is a reinstatement, and the animal will start pushing the button again in a repeated manner. And that for 10 minutes. And we are still in the extinction phase. So what this behavior suggests is that what we're really measuring is the motivation, the desire of the animal to take that drug again, because the animal is under the influence of the drug. So actually, this leads us to a hypothesis of interpretation. During the acquisition stage, animals learn that the interoceptive stimuli of taking drug are associated to the fact that their behavior is reinforced by doses of drug. On the extinction phase, animals are not under this exteroceptive stimuli, but their behaviors are reinforced by the drug. So here, the interoceptive stimuli will end up signaling that their response will be reinforced by the taking of the drug. So actually, in the extinction phase, when you put them under the effects of the drug, you're going to trigger those interoceptive stimuli. And that's why the animal starts pushing the button again, because you're somehow giving the animal the information that the drug may be available. And as you know, and Sayed showed this, the perception of the availability of the drug makes us know that we will be able to get some drug, and this is a stimulus to induce craving. So how are we going to test all of this? I'm going to go quite quickly on this, but the first manipulation is a beautiful experience by Holly Wise in Baltimore, in NIDA. So there is a formulation of a certain cocaine called methiodide cocaine, whose properties are that it is stopped at the brain-blood barrier. So it just acts peripherally. And this is what we show in the slide. When subjects get methiodide cocaine, there is an increase of cocaine concentration in the blood, but no trace of it in the brain, compared to when you're using hydrochloride cocaine that goes to the brain. So you can see a high concentration in the striatum part of the brain. So here we're talking about the acquisition of the behavior and then the extinction phase. So during the extinction phase, he tests the animals with the methiodide cocaine, which is the one that is stopped at the brain-blood barrier. So the experience shows that there's a reinstatement with the methiodide drug, a little bit less strong than with the other kind of cocaine, but it seems that the stimuli of the drug itself seem to be sufficient to trigger craving, even though the drug does not get to the brain. So, well, this was quite a prediction of the interoceptive hypothesis, which is now validated through this experience. Another prediction of this interoceptive conditioning that is quite unexpected is that when we inject cocaine with in a naive individual, we should be able, if we repeat injections, even though we're in the extinction phase, to stop the effect of triggering craving by the drug. And this is what we did with this experience. We repeated the injections, and we saw that the effect on craving would be considerably reduced with the repeating of the injections. And actually, we're able to show in an experience where it's the animal himself which will press and trigger, there was an extinction, a very strong extinction of the cocaine craving. It was almost a total extinction. It was almost as if the animal wouldn't have received any drug. What you can see here is that the psychostimulating effects of cocaine are stable. So it's actually that not that we're tolerating the psychic effects of the drug. We also showed that this is dose dependent. There is an extinction irrespective of the dose. There is this reinstatement of craving, And another important point, as I was saying before, is that stress generates interoceptive stimuli, which may be confused by those stimuli generated by cocaine. Now, if you stop the induction of craving by cocaine, this becomes stress. So after the extinction, stress cannot trigger craving in that animal model. This is what is shown in this experience. As I was saying before, lots of things regarding conditioning are independent and, are dependent on the context. So this means that the extinction of the effects of the craving by the drug depends on the context. In other words, if you're doing this experience in a context A and then you change the context, you're going to lose the effect of extinction. So it may limit the applications. And I think that, according to me, one of the challenges today is to find deconditioning procedures which are regardless of the context. And I don't think that this has ever been achieved yet. Maybe we could think about it. So, actually, we know that in the reinstatement model, the triggering of the craving is related to the activation of a pathway that goes from the pre-limbic part of the prefrontal cortex towards the ventral striatum. In particular, the heart of the accumbens nucleus. What we must imagine is that, well, the individuals suffering from that craving, they're going to seek that drug again because that pathway is activated. So we thought, well, after the extinction of the priming effect of drug, let's see if we can observe an extinction as well of that pathway that was activated. So this is what we wanted to check, and we undertook two different experiences. First of all, we took a neuronal marker of the activation of the nucleus accumbens and the prefrontal part. So in this experience, we had two groups, and they both learned to self-administer the drug.
Ensuite, on les a séparés en deux groupes. Un groupe qui recevait des injections de saline de manière répétée et un groupe qui recevait des injections de cocaïne de manière répétée pour éteindre l'effet priming de la drogue. Et ensuite, pendant le test, on a séparé chacun de ces groupes en deux autres sous-groupes. Un groupe qui recevait du solvant, un groupe qui recevait la cocaïne pour la première fois, mais qui n'avait pas éteint les effets interoceptifs préalablement. Donc, du coup, chez ce groupe-là, on doit observer une augmentation du reinstatement. Et ensuite, l'autre groupe qui reçoit du solvant et de la cocaïne. Et chez celui qui reçoit de la cocaïne pendant le test, on devrait avoir une extinction du comportement. Et donc, au niveau neuronal, on devrait donc s'attendre que si on éteint l'activation de cette voie cortico-striatale, on devrait voir une chute d'activité chez le groupe CC par rapport au groupe VC. D'accord ? Et c'est exactement ce qu'on a observé. Comme vous pouvez le voir, chez le groupe CC, l'activation neuronale que l'on observe chez le groupe VC est fortement atténuée, même dans certaines régions totalement supprimée. Ça, c'est donc avec une méthode. Et ensuite, ce qu'on a fait, c'est on a utilisé l'électrophysiologie in vivo, où on a mesuré l'activité. Donc, pendant que les animaux sont en train d'être testés, l'activité neuronale dans le cortex prélimbique du cortex préfrontal. Donc, chez les animaux qui n'ont pas éteint le comportement, on a une forte activation de ces neurones dans cette région. Et après extinction, on a une diminution de cette activité en réponse à la cocaïne, démontrant que l'extinction des effets comportementaux de la cocaïne est bien associée à une extinction de l'activation de la voie neuronale cortico-striatale. Ok, donc 0, mais je vais m'arrêter là. Donc, petite conclusion rapide. Donc, l'ensemble des données suggère effectivement qu'il y aurait une voie d'activation interoceptive d'activation du craving qui impliquerait les effets interoceptifs de la drogue. Également, tous les effets interoceptifs d'autres événements émotionnels, stressants, etc., qui reproduisent ou qui sont confondus par l'organisme avec ceux de la drogue. Voilà, je vous remercie pour votre attention.
Merci, Serge. Merci, Serge. Bon, on va passer
thank you, Serge. Now we're going to answer potential questions from the public or the panel. Do you have questions? Okay, so rather we're going to have all questions at the end. Right, so all questions will be summed up at the end. And I'm going to give the floor now to Louis STINUS. Is going to highlight the contextual drug conditioning, the appetitive and aversive effect of withdrawal. Louis STINUS, you have the floor. Well, I would like first to thank my thinker, Archives, Jean-Pierre and Marc AURIACOMBE, who invited me to give this presentation. But I would also like to thank all the members of IBICIA for the organization of this wonderful Congress. So you're all familiar with the cycle of addiction. You know that moment of sort of honeymoon, but then when you continue with consumption, then you lose control and you start with abuse, and then you become dependent. After a while, you try to abstain, but the rule, unfortunately, is that you have a relapse. And why is that? Well, when dependency is being created, there's a neuroadaptation at the level of the entire body when the subject consumes for example, drugs. And relapse can be activated or provoked by a number of factors, one of which being stress, and then the conditioning effects. And I'm going to talk about a number of results that we collected all along my career at the CNRS, which is the French Research Center, National Research Center. Before I continue, I'd like to give you two examples that Chuck O'Brien would always mention. He would tell the story of a patient of his, a former heroin user, who had stopped using heroin, was reinserted in a society, and one day, because of his for his job, he had to travel to New York, went through a metro station where a few years before he had a drug injection, and because of that, he started using heroin again. He was also mentioning GIs in Vietnam who would be consuming lots of drugs, and for many of them, not all of them obviously, but many of them, going back home was accompanied with stopping using drugs, so returning home to the States, I mean. Hence the high impact of conditioning. The first experience I'm going to talk about is very simple and quite obvious. The place preference for heroin injection. And I'm going to show you the protocol. This is a cage with two compartments. two cubicles. The space is exactly the same. The setup is slightly different. The paper wall, so to speak, is different, and the carpet is different as well. It's not a carpet, but anyways, the flooring is different. So in the first days, the animal can freely roam both rooms. Then we close the door in between. And so the solvent is going to be injected, for instance, here on odd days. And the other days, we inject a very low dose of heroin in the second part of the room, in the other cubicle. Then on the test day, we open the door and we look at what's happening. You see that on the following day, so after the conditioning phase, there's a very strong place preference. The rat is going to spend a lot more time in the part of the room where they received heroin. The heroin dose that we used was extremely low. In fact, the rat receives 25 micrograms of heroin, which is really the minimal amount to have a psychostimulant effect. But 2 months later, the rat still remembers that he had a nice experience and that drug injection was very agreeable. So 2 months after that. The second protocol I would like to talk about is behavioral sensitization induced by repeated administration of nicotine. So this is a phenomenon which was not mentioned by a number of players in tobacco addiction. You have here different groups of rats. Every day they're going to receive an injection either of a solvent, so doses is 0, or nicotine, 1, 0, 1, 2, or 4 milligram. So you do see a behavioral activation. So we measure the locomotor activity of rats, but already at 0.2 milligram, you do see a sensitization, which is of course a lot obvious when you inject 0.4. And I'm going to show you how this sensitization is only expressed in the context in which the rodent received the nicotine in the first place. So it's place dependent. Now, this is the this is a setup. We have two environments: activity cages, so cages where they have activities. There are ramps of photoelectric cells, which are going to provide us with information about the locomotor activity. And then we have a breeding cage, where the animals have no activity, and rodents are used to being placed in both cages. Every day for 7 days, we inject 0.2 milligram per kilo of nicotine to those rats. It's a very low dose, once again. We have three groups of rats. Every morning, those rats go in the activity cage. After half an hour, when they sort of slow down, the nicotine group is going to receive an injection of nicotine. The nicotine group, the other group is going to receive a solvent, and then the control group is only going to receive solvent. 2 hours later, when the effects of the injection has worn off, the rat is back in the activity cage, and this is when we inject the second injection. The nicotine group is going to receive nicotine, the solvent group is going to receive solvent, and the control group is going to receive solvent as well. So you see that you have two groups of products with exactly the same pharmacological experience, but in different contexts. So we're going to repeat that procedure for 7 days, hoping to induce an increase of sensitivity to nicotine, and then comes the test phase. So 24 hours later, we have a test with an injection of solvent. So we want to check whether there's a placebo effect and look at what's happening. Yeah, here we go. Those rats which are used to receive nicotine in the activity cage, when they receive a solvent injection, they answer in the same way as if they had received nicotine. The control group, they never received nicotine in the first place, they behave like the animal receiving solvent. But this group, they have the same pharmacological experience, if you think about it. But they do not have a placebo effect because their experience of nicotine is not in the activity cage, but in the regular cage, in the breeding cage. The next test is the following. We inject a very low quantity of nicotine to all rats. So all rodents receive nicotine, and we try to measure the ones who are sensitized and the others who are not. So despite the fact that they all received the same numbers of injection, we should have a higher sensitivity in those ones, which is not the case. So in yellow, you have the control group. For the first time, they received nicotine. The second group, those were rats who were already sensitized to nicotine, but in the regular cage. And the third column here is those rats receiving nicotine in the activity cage, and so they receive nicotine once again. So there's indeed an effect. We increase the sensitivity, and this is context or place dependent. And this is clearly shown here. Now, those were what we call appetitive effects. Now we're going to talk about aversion. So we can also condition an aversion. You can create an aversion in a specific place in rodents. So those are the contraptions in which, or rather the setup in which we create that conditioning. So there are 3 different places. The flooring is different. The wallpaper, so to speak, is different. And again, we have cells in that entire setup in order to measure the activity level. So those rats are made dependent by an implant, by small, slow release morphine implants that were provided to us by NIDA. So we implant those implants, and we are going to inject 15 microgram per kilogram of naloxone. This is a very, very low dose, but the intent here is to create a withdrawal effect. There are no physical signs of withdrawals, so no diarrhea, no increase saliva. So you cannot tell that the rat is in a state of withdrawal, but we are starting to induce withdrawal. So we place the rat in the setup, and the rat can spend his or her time wherever they please. But after a while, we are going to be confined or locked down in one of the different cubicles. So again, we inject naloxone and we lock down the animal in each of the different cubicles. At the end of the test phase, we remove the dividers, and the rat is going to choose the place where they, I suppose, feel better. Now, so this is the place aversion after that conditioning, so 24 hours after that conditioning phase. But look, 54 weeks later, so that's 1 year later, the aversion for the place is still there. The rats can still remember that they felt bad in one of those places. And this was done obviously with different groups of rats. So the rat tested at 54 week spent 1 year in a regular cage, not receiving any injection whatsoever, but it's just a memory of something which happened before, but 1 year before. Now, I'm going to talk about something different, about withdrawal, connected to withdrawal and connected to nicotine use. I'm going to talk about the tobacco paradox. Tobacco use is one of the most intense form of dependency. However, nicotine has very low reinforcing or reinforcement effects. When you think about it, the nicotine use is very specific or particular. When a smoker inhales nicotine, in a few seconds, nicotine reaches the brain. That's exactly the same effect as if you had intravenous injection of nicotine. Now, when you have one cigarette, 10 inhalation, which means 10 injections of nicotine. One pack of cigarette per day, 200 injections. Per week, 1,400. Per month, 5,600 intravenous injections. There is no other drug in the world with such a high frequency of consumption. However, nicotine is not the only substance present in tobacco smoke. There are other substances in cigarettes, and one of the substances is very interesting. And we observed in smoker a decrease of about 40% of the activity of monooxidase in regular smokers. So I'm going to explain in more details now. So this is a publication by Fowler in 1996. This is a brain MRI. So the MAO in the normal individual, and this is a brain imagery in a regular smoker. This is a reversible effect. A couple of weeks after you stop smoking, your brain is back to normal, so to speak. But all this to say that tobacco smoke contains inhibitors of MAO, and obviously the tobacco industrialists, it's not them adding something. If you burn tomato leaves, because it's the same family as tobacco, if you burn tobacco leaves, when they reach pyrolysis, you do have the formation of the same molecules. So in smoke, you do have nicotine, but you also have IMAO, and they are also present. And you will see that AMAO, are here to reinforce the effects of nicotine. Oh, I'm back, says the speaker. Now, in order to prove that, we try to measure the state of withdrawal in rats, which are only nicotine dependent. And you're going to see that there's indeed a conditioned aversion, but this is only a transient effect, contrarily to what I showed you with morphine. Those rats dependent on morphine have a long-lasting dependency. So how do we make those rats dependent? We implant mini pumps. They are about the size of a bean. And so we implant that under the skin of those rodents. And those pumps, osmosis pumps, are going to inject that quantity of nicotine every hour for a duration of 14 days. Now, this is the same setup that I already showed you. So we're going to associate one cubicle with an injection of solvent, another cubicle with an injection of mecanylamine. Mecanylamine is an antagonist to the receptors of nicotine, and this is going to create a small withdrawal syndrome. Now, those are the results. If you test rats which are only nicotine dependent, and in which we provoked a state of withdrawal. So if we test those rats one day after the conditioning phase, there's an obvious aversion. But that aversion disappears after 1.5 months, which means that nicotine does not have the same addiction effect as morphine, for example, and the withdrawal of nicotine is not very violent. However, what happens when we try to mimic the smoker situation? So we make the rat dependent on nicotine and to inhibitors of MAO, as if the rat was effectively smoking tobacco. So we try to measure the place aversion when the dependency was created by nicotine plus IMAO. And so the pumps that we're now going to use are going to deliver nicotine and IMAO. So same protocol, those are the results. That all of a sudden, we realize that the conditioned aversion to a place, which appears on the following day, 224 days afterwards, so 8 months later, you still have that place aversion. Those columns here represent the summer months of that year. And those months are very particular for those rats. Now, when you associate nicotine plus IMAO, you reinforce the intensity of dependency and therefore the intensity of the withdrawal syndrome when you stop smoking. I'm almost done, I'm sorry, says the speaker, and I pressed couple of times too many. Anyways, so IMAO work in synergy with nicotine. They increase the reinforcement effects of nicotine and also the motivation of smokers, their motivation to smoke, and they also reinforce craving. They also increase the intensity of dependency to nicotine. They increase the negative effects of the withdrawal, and therefore they increase the intensity of conditioning of those subjects. And I would like to thank you for your attention. Now we're going to ask the next speaker to take the floor.
Now, we're going to hear the presentation of Fuschia SERRE and Marc next to me. Are there any conditioned effects in humans? What are the data and what are the research and clinic implications? Thank you. Indeed, I'm going to talk about the conditioned effects in humans and what are the implications for research and clinic. These conditioned effects are going to be the basis of development of the paradigm of cue reactivity, stimuli reactivity, which are the stimuli associated to use, the capacity to trigger responses even in the absence of a substance. There can be, the stimuli can be objects, when they're going to acquire the capacity to trigger response or reactivity without the presence of the substance. This reactivity will then motivate the individual to consume again. We call that condition stimuli or cues. The paradigm is the following. The individual is in an experimental room. They are presented two types of stimuli. First of all, a neutral stimuli, and then stimuli related to substance. And we're going to compare the responses, the two responses. What do we see? We see a subjective reactivity, self-reported reactivity, the most frequent one being an augmentation of the craving. The craving is a desire to consume, but it's a persistent desire, which is pathological. even it's in its intensity, and it's not voluntary. We also observe a physiological reactivity with an increase of the heart rate, increase of the skin conductivity, a decrease of the skin temperature, and a change in the cortisol response, which can be measured in the saliva. We also observe brain reactivity with the activation of some areas of the brain, as we can see on this image. Regions involved in several functions of visual, memory, learning, the reward system, and we also observe a change in the functional connectivity between different regions. These results highlight how the paradigm and the reactivity in humans can inform us about the neurological conditions. Meta-analysis show that the results of these studies show the more important effects. An analysis on more than 128 studies to study tobacco addiction. Among the different types of reactivity, the largest one is the increase of the craving. It's the most important, more important than the physiological reactivity. In this meta-analysis, the only physiological reactivity is the skin conductivity. A limit often reported is a difficulty to bring together the results from very different analysis with different populations. A review lists all the different modulators of reactivity. They can be classified according whether they're related to the individual or to the experimental procedure. Among those that are related to the individual is the severity of the addiction, the length of abstinence. They will have more reactivity, and this is to be related to the incubation. There is an increase of the induced craving during the first weeks of abstinence. And now the modulators is related to the procedure, the way you present the stimuli. In this tobacco analysis, the reactivity with images was greater than the reactivity obtained with virtual reality. Another factor which is very important is the complexity of the stimuli. Indeed, in the lab, we use objects, but in the natural environment, stimuli can be very complex and different and context dependent. Can be sensations, feelings, smells, can be diversified. And we can imagine that it's not only one stimuli, but an association of several stimuli. We can think that the sequence, the temporal sequence of the stimuli is also important to trigger a reactivity. It's interesting because in this meta-analysis, we see that the observed reactivity in daily life is more than the reactivity observed in the lab. So this is why it's interesting to study this phenomenon in the daily life in the natural environment. This can be done with the IMAO methodology, where we assess individuals in their daily life with a questionnaire on their smartphone. You can assess people in their natural environment to have a good ecological validity and to study the influence of the context, however complex it is. The measures are carried out in real time. There are several questionnaires, so you can see the different biases. In the lab, we have used this methodology. So we have highlighted the cues craving usage model. This is a study with people who were we were at the beginning of the treatment for substance abuse. An increase of the intensity of the craving at tea time was associated to an increase in the probability of having a usage in the next hours. The craving explained the usage and not the opposite. What we are interested in today is more the fact that we've observed that an increase in the number of cues of conditioned stimuli present in the environment is going to be associated to an increase in the intensity of the craving in the next hours. We compared two types of cues. First of all, the standard cues, they're stimuli normally associated to the substance or its use, for example, a bottle for alcohol or an ashtray for tobacco, and individual cues, stimuli that the individual personally associates to his use. Can be a specific place, a feeling, a sensation, an activity. And what we are showing is that when we measure these individual cues, we see a stronger effect, an extended effect on the craving compared to the reactivity observed with standard cues. So what is the implication for the addiction treatment? So we're going to help the patient obtain and maintain abstinence, at least a significant decrease in the use. So we can help the patient in managing the craving, decreasing the craving by pharmacological treatment or by developing strategies to better manage the craving. But we can also act on the conditioned effects, and especially we can help the patient in identifying the cues and helping in avoiding them. At the lab, based on this observation, we have developed an application. It's called the Craving Manager. It's a smartphone application for treatment. It's going to use the EMA methodology to propose to act on the craving as soon as it happens and in a personalized way. The application is going to try to anticipate the craving to prevent it. For that, the application is going to assess the presence of this conditioned stimuli in two ways, either by asking the individual or in a passive way. For that, you can use the geolocalization to identify geographical areas that are associated with a craving or to prevent the craving. We can also add connected bracelets to measure the physiological reactivity. There is a thesis currently going on to identify the physiological response associated craving to trigger interventions, even if the individual is not aware of the appearance of the craving. So we just saw the conditioned effects to predict the use and the relapse in the next hours. In fact, these conditioned effects are also going to be interesting to predict a long-term relapse. That's we've highlighted in this study. The study was conducted with patients that were opium addicted. They had been in treatment and stabilized patients, so they received this procedure in the lab. The different reactivity parameters were measured: psychological reactivity, euphoria, and also the physiological reactivity, including the measurement of cortisol in the saliva. We observe. Then there was a prospective follow-up to see what happened to them in the next 3 months and to see whether they're relapsed. We see that indeed the relapsers during the 3 months had higher reactivity during the experimental procedure, especially in the subjective reactivity with more craving and euphoria, but also in physiological reactivity in the cortisol response that was measured in the saliva. So the reactivity is a prognostic marker for the risk of relapse in the next months. So it's important to develop procedures to reduce and extinguish this reactivity. This is the case in the extinguishment procedures that were developed in the animals. So we expose the individual to this stimuli repeatedly without reinforcement to extinguish such response. But we see that results are mitigated. So we wonder whether it's not due to the type of stimuli presented. This is why other studies decided to go further and to use the virtual reality. In this case, we're going to present environments. The individual will be able to navigate in these environments and to interact with them. We see in the images environment related to the use of tobacco when you're in a traffic jam or when you're in a waiting for a bus. So we can wonder whether it wouldn't be through this extinguishment procedure. They take place in the lab, and the effects are relatively low, and they don't last in time, and they don't persist outside of the environment. Now, some authors offer more techniques of augmented reality. There you represent the daily life is going to be enriched by the projection of virtual elements in real life. This is still at the preliminary stage, but we realize that this type of procedure makes it possible to induce craving, and this could be a promising avenue for the future. Now, concerning this brain reactivity, we propose to use a neurofeedback. In this case, neurofeedback consists in presenting stimuli and to record the brain activity, then to present in real time this activity as a sound or in a visual way. And the purpose is to train the individuals to regulate this brain activity with the objective that if they can train and can regulate their brain activity in these regions, then they can reproduce such modulation in their natural environment. Again, for the time being, this is at the experimental and preliminary stage, but the first studies show that this technique makes it possible to reduce the brain activity in the concerned regions, and it's associated with a very promising reduction of the craving. But now we have to conduct studies in larger samples, and we have to see whether the effect continues over time. In conclusion, these condition effects have an important role in experimental research, and they enable us to better understand the relapse mechanisms and neurobiological relapse substrates. We saw that it's interesting to get out of the lab to develop research in real life conditions. And these condition effects are used in addiction treatment, but it's also going to give a new promising approach for addiction treatment. Thank you. To help in the debate, let me say that we often forget that the reward system exists because there are substances which are secreted by the body and with the endorphins. If you don't have endorphins, you have no reward. So you have to take that into account because it's an essential point. When you talk about heteroceptive, you have to take into account the power, the secretion power of the of these products. This is important to understand the situation. This is why your presentation was so interesting. Thank you. First of all, it's a great presentation. It's an opportunity to see over the generations the development of research on addictions and conditioning. So this is very interesting. I have a question for Serge AHMED and Fuschia SERRE. In the presentation reports that conditioned stimuli, you have the general stimuli and personal ones, individual stimuli, and these conditioned cues are more powerful than the general cues. And in Serge AHMED's presentation, he insisted on the conditioned stimuli interoceptive versus external ones. And I think that they were more related. My question is the following. Is part of the individual condition cues that were highlighted, that were shown in humans, could be partly heteroceptive cues acquired by the individual over time? Absolutely. The difference between the standard cues and the individual cues is that the individual cues are cues could be standard, but we just ask each individual what were the stimuli, the cues for him. So there should be a match between the two. When we ask the individual, we see that the individual stimuli can be very different. There can be sensations, smells. So it can be also something interoceptive. Can we invent exteroceptive cues based on the knowledge of each individual? For instance, the smell, what you explained, that was very clear. In your method, in a treatment, do you take that into account? The singularity of each individual. Exactly. And that's what's important in the extinguishment procedures. It's personalized medicine. For each individual, you have to find what are his or her stimuli which are different from one individual to another. That's why it makes it difficult. Thank you. That's what I wanted to hear. Another question? This presentation reminds me of something. It reminds me of Proust's Madeleine. It's exactly the same thing. Thank you for your words. Any other question? I have a question for Fuschia. You insisted on the difference between what you call the lab context and the ecological context, which is the everyday life. So you said that the craving induced in the lab was less intense than in a more ecological context. So you said it was due to the fact that the stimuli were different, stimuli which are very personal in the ecological context. In the lab context, they're more generic cues. But there is also the dimension in my presentation of the context. I wanted to know what you thought about the role that in all the associations are context dependent. And even if you have a personal cue in the lab context, because of this change in context, you will have a decrease of in the effect. What do you think? As you said as you showed in your slide, the substance is potentially available, can induce craving or change it. So we can imagine that the craving in laboratory is completely different than the one in real life. I would like, before asking a question, I would like to thank you all for these great presentations. In our mind, when we drew up the program, we thought in the inaugural session, we would first talk about the power of conditioning. In practice, in our clinical practice, I see that if you don't take into account the power of the conditioning, when we observe the patients, we see that, especially for the patients where the physical dependence is less in cocaine addicts. We see the extraordinary pregnancy of the environment for some cocaine addicts that are extremely dependent. If you put them in a hospital system, in a place where they don't have any cues, they say, "Oh, it's great, I don't have any more craving." They go to a consumption area and everything explodes. So for all my patients, they all know the experience that you were mentioning of the rat after self-administration of cocaine. 1 month after with the passive injection, everything starts all over again. It's very important to see how much this you have to take the power of the conditioning. So you have to think of all the tools, whether psychotherapeutic tools or pharmacological tools or others. And you have to implement all these tools. So it's important to say all this. Luis reminded us that for nicotine addiction, the power of the potentialization of the conditioning by monomyoxidase. So we see that also in a clinical setting, because the relapse of smokers occurs a long time later. In France, Marc said that the nicotine substitution is not extended enough. It should be longer, not 1 month, but much more. Couldn't we imagine a pharmacological agent that could limit the impact of the smoked tobacco, that could limit the inhibition of monomyoxidase? I don't know who that question is for. You're asking a question which it's impossible to answer. Yes, substitution by nicotine doesn't cover everything. And as you saw, nicotine alone are associated with a monomyoxidase inhibitor. These are two different worlds. I don't know. Maybe associated. You can associate nicotine with the weak inhibitors of monomyoxidase and decreasing the dosing gradually. I don't know. This is a magical session. This is exactly what I expected from the session. I had seen the topics you wanted to address, but it's great. What you told us is made me very happy.